So good morning, everyone. I hope you still have a little bit of energy left for this last morning. So I'll be talking about how we can get a little bit further than just describing our tissues in terms of which cells there are and where they are located and what kind of spatial neighborhoods they're in, but really go one step further than just describing the components of our tissue and really look at the interactions, right? How do cells interact with each other to implement the functionality of a tissue? So that's going to be the topic of today. And if this is working, yes. You've seen, of course, all these amazing technologies that are advancing very rapidly. What is important for us today, if we think about cell-cell communication modeling from this type of data, is, of course, first, if the technology that we're using is up to single-cell resolution or not. Some of the technologies really allow you to, for example, by segmentation, look at single cells and then assign transcripts to these cells so that we can have an idea what's the spatial location and the expression distribution of the transcripts at single cell resolution. But other technologies are a bit larger spots covering multiple cells. You can still do cell-cell communication analysis on these, let's say, larger regions, but then you're just reasoning at, let's say, a higher level, not at the individual single cell level, but at region level. There's also the second important aspect of these technologies is whether they are targeted or not. So in the targeted technologies, you have to design your own panel, which basically means you have to know very well the tissue that you're working on, right? Because you have to have all the right markers for identifying all the cell types that you want to study. And if we're specifically thinking about cell-cell communication modeling, you also have to put all the ligand and receptors in your panel if you want to study cell-cell communication. Some of the early data sets that were published were totally not suitable for cell-cell communication analysis because the marker panels didn't just cover the necessary ligands and receptors that you need if you want to study cell-cell communication. Still, there are some bypass things that you can do, inferring if you don't have these ligands and receptors by looking at some downstream pathways. But it's much easier if your panel is well thought about in advance for the specific biological question that you want to answer. And that is still, I think, something we're missing a bit in the field because people are just putting out data sets and only a posteriori ask the question, what can I now do with this data set? Instead of the reverse question, which is what is the biological problem or the biological question that I want to solve, and then generating the appropriate data set for that, right? So that's very important. What's the biological question that we want to answer? And that will determine basically all the choices of the technologies, whether it's a targeted or not panel, and so on. If you have the choice, right, an untargeted panel, which basically measures very broad expression of almost all genes, is a little bit better for exploratory analysis because it will allow you to measure, for example, all the ligands and receptors, downstream genes of pathways, and so on. And then we also have the different modalities. We have transcriptomics technologies. We have proteomics technologies. Both of them are very interesting. And basically, if you have both, it's even better if you want to study cell-cell communication. And the first technologies are now basically appearing where you can combine transcriptomics and proteomics on the same slide. You can always do consecutive slices, right? If you have one slide with a spatial transcriptomics, next slide spatial proteomics, but then again need some kind of fiddling to unify or combine these two slides. Would be better if you have all the modalities on exactly the same slide, but that's still technically work in progress, right? Okay, so what are we interested in going to towards the future? So you know that every new technology that comes out in the single cell field is just basically, again, a next generation microscope, right? You measure more, you measure deeper. So you have more and more data, and you need to combine all these different types of data to, again, answer this biological question. So we have what we call now the research direction of functional spatial omics, where we measure different modalities, but we, in addition, want to also model all the interactions of all the components that we measure, all the genes, proteins, and so on, in their spatial context, and also by looking at all the mechanisms that are really determining the cell state and functioning. For example, by looking at models of cellular interactions and models of gene regulation. And as I will show you later, these actually need to be in the future combined if you want to make the most out of the data. So here you see an example of a spatial transcriptomics data set on melanoma that we published last year. You clearly see all the different niche environments here. This is a spatial proteomics data set on the liver. So it's one of the first spatial proteogenomic atlases that we published already a few years back in Cell. So I don't know if there's anyone who recognizes this picture. Yes, this is a kind of typical promo picture of the Single Cell Genomics Conference, right? The idea is that we have all these technologies that measure all the parts, but now we want to see how the parts actually interact to model the whole system and learn how the system as a whole is functioning. So what is very important, I think, is that you take a little bit of time once you have such a data set to really look into the quality of the data. So this is an almost quote of one of my favorite metal bands. I don't know if the young people still know Metallica, but they have this famous song, Nothing Else Mattered. But if Metallica were bioinformaticians, then they probably would have called this song a slightly different title, and just tells you that you really have to take the time to do quality control and pre-processing, especially in spatial omics data sets. Now, the unfortunate thing is that these technologies are so new yet that we don't have any standardized pipelines yet to process spatial omics data. So basically, we're still in the research phase, right? We're still finding out what are the artifacts that are happening in this type of data for the different types of technologies, and what kind of QC and pre-processing steps do we need to design to really get to high quality data. Because if you do very good pre-processing and QC of your data, that will have a huge effect on any downstream analysis that you're interested in. You could get to quite different biological conclusions if you do a decent quality control and pre-processing of your data. So that's why it's very important that you spend some time on that. Oh, sorry. So there are different steps in this pipeline. I'm not going to go too much into detail. You have seen this already in this previous lecture, but we recently published a new pipeline that is specifically designed to help people fine-tune all the parameters in the different steps of these image-based transcriptomics data sets. It's especially very good because it gives you a lot of QC metrics and plots that tell you which part of the tissue are good quality. For example, if you plot your transcript abundance here on your tissue, you can see that in certain regions quality is very bad. You can decide to leave out these. You can look at the different cell types. You can look at distributions of your transcripts across your cell types. And all of that can actually be used to do the QC on your data. So it's an interactive pipeline, right? So biologists can actually use it in a very user-friendly way. They can look at regions of interest, play with different parameters that, for example, influence your cell segmentation, and you can choose the best parameters for your cell segmentation. And that has a huge effect. For example, what you see in the top picture here is if you run a standard vision pipeline on this data set here, and then below, if you use the spiral pipeline, where you actually do the optimal parameter tuning for all the different steps. You can see there's huge artifacts here where certain regions and certain cells in this region are not well segmented. If you fine tune the parameters, you're going to actually segment these cells here. What you can also see is that if you then do downstream annotation of these cells here, that, for example, for this pipeline here, all the cells that you see in blue here are basically annotated as unknown by the cell annotation algorithm. You see that in this case here we have 23% of unknown cells, as this big blue blob here. If you do the better segmentation here, you do all the pre-processing steps for the imaging and the segmentation, you end up with only 4% of unknown cells. What is even more interesting is that some of the cells you will actually miss. Some specific cell types you might even miss in this first standard pipeline here, while some of the cell types you only recover well if you fine-tune some of the parameters of your algorithm. So I'm not going to go too much further into these pre-processing details. We also contributed to this spatial data format, which is kind of a data structure for storing and allowing you to work with this type of data. It's also designed for the future integration of different consecutive slices and so on. So if you want to read more about that, this is the paper. All right. Where do we want to go with all these technologies? Basically, we want to move into the field of next generation pathology. So you take a tissue from a patient, you actually, this is the classical pathological stain where people are, for example, interested in certain spatial patterns, for example, some types of cancer growth here. Here you see two examples, replacement growth and desmoplastic growth, which are two different spatial patterns that you can see in tumors. And basically, it's very important to distinguish these because patients with one type of the other have more survival probabilities than the other. Now, if you go from these classical stains to what we call next generation pathology, so this is a 200-plex spatial proteomics example that we did using the Maxima platform. So that will give you a huge wealth of information for all the different cell types. Now you can look at single cell resolution. You can see this kind of region here at the bottom left, where you have these red colored cells here, which are profibrotic macrophages. You have the green ones in this region on the top right here, suppressive macrophages. You can study, for example, the signaling between all the different cell types in these different regions. You can think, of course, that could lead to new types of biomarkers, but also to new types of treatments if you better understand what is going on between all these different cell types here. And of course, then you can, from these segmented cells here, you can derive spatial statistics. You can compare distributions of, for example, spatial locations of these cells. You can apply all the spatial statistics that Mark covered yesterday. And in this way, you can get to more quantitative image analysis. And so that will be very useful if we want to make more out of image analysis in general. So it's not necessarily limited to spatial omics technology. You can even do this on normal confocal microscopy images, for which you have a lot of data already. All right. So let's get to the core of this lecture. So the cell-cell communication modeling. So let's start first with some basics of cell-cell communication. So there exists, of course, a plethora of different ways that cells can communicate with each other. For example, cells can regulate themselves, called autocrine regulation. You have cells that are touching next to each other and have direct contact with each other. You have dendrites and nanostructures. You have paracrine signaling through, for example, vesicles. You have also paracrine signaling through soluble factors. For example, some sender cells producing some ligands that diffuse. Some receptors on receiver cells are receiving these ligands and are then putting into action how downstream signaling cascades. And you also have endocrine signaling, where you have longer range communication by, for example, taking care of a bloodstream or other channels in your body to have connections between distant tissues here. Most of the tools that I will focus on today will actually look at how we can study cell-cell communication from transcriptomics data. Now, that seems logical to you maybe now if you read all these papers, but when 5 years ago we talked to biologists that we wanted to study cell-cell communication based on transcriptomics, everybody laughed in our face and said, ha, you need proteomics data for that, not transcriptomics data. And that's, of course, perfectly right. But transcriptomics data is much more abundant, easier to get. And so a lot of tools were developed to see if you could infer cell-cell communication from transcriptomics data. So we have all the cells, either from single-cell transcriptomics data or from spatial transcriptomics data. We want to infer which type of sender cells are sending which type of messages to receiver cells. In some cases, we really want to study cell-cell communication between individual cells. And most people sometimes think that this is the logical thing to do, but you have to also think about the signal, right? You have much more signal if you pool cells into cell types. And so basically many tools still study cell-cell communication by looking at groups of cells, typically grouping cells into cell types and then check how different cell types communicate with each other. But in reality, of course, cell-cell communication happens at a single cell resolution. It's not because cell type A communicates to cell type B that all cells of cell type B are kind of reacting in the same way to kind of messages from cell type A, right? And there are now also quite a number of cell-cell communication tools that really allow you to study cell-cell communication at the individual cell level. But that's inevitably going to always be much more noisy than looking at groups of cells, right? You can specify these groups in many ways, right? You can cluster cells into cell types, but you can also look at spatially related groups of cells. And of course, if you have spatial transcriptomics, you have one layer of additional information, and that is that you know that if cells are located near to each other, there's just a higher probability that they will be communicating with each other. So all of that can be combined, of course, to improve cell-cell communication modeling. All right. So what I'm going to describe to you today is how we basically can model these type of cell-cell interactions. And I'm going to focus here on ways to study functional cell-cell communication. What do I mean by functional cell-cell communication? Let's say, for example, I ask Ahmed, how did you sleep tonight? So that's a functional cell-cell communication because he responded, right? I know that there was an effect of my communication because he heard me and he responded. If I would say, for example, Helena Crowell, did you have a whiskey last night? Then still we're communicating. It's a bit more long-range communication. Took maybe a little bit more time for me to get feedback from Helena, but still she was functionally communicating with me, right? Same is happening with cells, right? If you have a sender type A that diffuses some ligands, some cells will respond to it, others will not respond to it, depending on the environment and also the internal state of the cell. All of that kind of impacts how or what is the effect of cell-cell communication. So let's have a look at what are all the components that we actually need to model if we want to study functional cell-cell communication. So I'm going to focus here on this paracrine type of signaling where we have sender cells that are producing some ligands that are then received by a receiver cell through its receptors. So cell 1 is our sender cell. It's sending a message to cell 2 through a ligand receptor interaction. There can be different ways, right? So the receptor can be a surface receptor or it can be an intracellular receptor. There are different technologies that you can use to measure that. But basically, it's producing a message. The message is received somewhere in the receiver cell. Then you have some downstream signaling cascades. For example, a lot of correlation and other types of events until you finally reach some transcription factors. These transcription factors are then putting into action how downstream gene regulatory cascades of primary and secondary transcription factors. It could be a whole gene regulatory circuitry here behind. As a result of that, you have some primary targets, some secondary target genes that are maybe changing their function and can be measured by looking at their expression profile. So in general, some target genes that will be affected by our cell-cell communication. And then maybe these target genes are maybe producing some ligands that maybe can be used as a feedback mechanism to our original sender cell or to another cell, putting into action a whole downstream signaling cascade between cells, or could also upregulate some receptors that would then make that this cell too kind of can receive other types of messages. There could be a whole complex intercellular interaction dynamics that is really happening due to this first interaction effect here. And basically, if we want to study functional cell-cell communication, we need to look at the whole pathway here. That's basically what we want to model, right? And that's what I said in the beginning, where it's not just looking at cell-cell communication, right? It's also modeling all the gene regulatory networks, because these are driving how this cell too will respond. And you need to know all the mechanisms that are happening in these cells here. So what types of data do we actually need in the ideal case, right? Suppose we want to study the full mechanism here. We would need data for studying the ligands, which can be cytokines, proteins, metabolites, whatever, right? So you need proteomics, metabolomics, imaging to study the ligands. So you need to also have ways to study the receptors. Could be also proteomics. Could also be flow cytometry data if you're looking at surface receptors. Could be site-seq. I don't know if you've ever seen site-seq. It's a combination of transcriptomics and surface proteomics readout. Very nice type of data. Imaging data, also imaging flow cytometry can be an interesting technology. Then you need ways to have all the downstream signaling here. Typically, it's a lot of protein-protein interactions. Then you arrive onto the gene regulatory networks. So for that, you need transcriptomics and attack-seq, chip-seq, every kind of bit of data that can really help looking at transcription and translation machinery here. The target genes you can also measure through transcriptomics. And then basically, we've almost covered everything. We still want to have additional information on where the cells are positioned in the tissue. So that's where we have all the spatial versions of these technologies coming in. And then there's one last aspect that we're missing, and that is basically that the whole communication process, just like communication between us humans, is basically a dynamic process, right? So we have messages, we have responses, coordination. So we also want to look at the dynamics of the whole process, right? You basically want to see what are the whole conversation pathways between cells. And you also want to model that. Of course, you need time series data for that, or perturbations like perturb-seq, CRISPR type of treatments. Maybe you block some receptors, and that can be kind of a perturbation that you see. You want to see what kind of effect that has to infer some causal knowledge about your system. Also, a very nice technology for dynamics is intravital imaging, but it's very low throughput. You can only look at a few proteins simultaneously, but interesting dynamics of interactions that can be modeled. All right, sorry. So if you talk to people in natural language processing, basically, if you have a text, you want to see who's the subject, who's the what is happening, where to whom, and so on. The same we can actually try to study in cell-cell communication. We want to know which cell is talking to which other cell, how, why, when, where, and so on. So we want to actually study the language of cells. And basically, if you think about natural language processing, you have seen very big changes in AI models, like all the ChatGPT type of models, which have usually boosted the capabilities of language modeling, right? You can imagine that similar things are going to happen in the single cell field, right? We want to study large cellular language models, but first we need all the right technologies to measure all the components that we need to study that. So let's start with some of the most basic types of cell-cell communication tools that are tools that basically study interactions between ligands and receptors from transcriptomics data. So what's the basic recipe for these tools? And these are tools like CellPhoneDB, CellChat, and so on. Many of these tools are implemented into larger frameworks like Liana Plus, which I think Francesca is going to demonstrate to you in the next practical session. So basically what these tools are doing is, first you have your gene expression matrix, right? All the genes in all the cells. What we're going to do is we're going to filter a gene expression matrix for only the activities of the ligands and the receptors. Typically, you use some databases to filter these out. So then typically, you're going to aggregate the expression level across all cells of a particular type. That's what I said earlier when most of these tools actually study things not on the individual cell level, but at the cell type level here. So most of the tools just aggregate at the cell type level here. Could also not aggregate, but then you have much more noisy measurements, right? And then for each possible pair of sender and receiver cell types, what you're going to do is you're going to look at all possible ligand receptor combinations, and you look at your expressions of the ligands and receptors in the sender and receiver pair of interest. And you're going to combine the expression profiles to calculate some kind of a communication score, right? Basically, if your ligand has a very high expression, your receptor has a very high expression, that ligand receptor pair between that sender and receiver cell type will get a high score. And basically, you're going to do that for all pairs of cell types and per pair of cell type for all ligand receptor interactions. Of course, you can do more fancy things like take into account cell type specificity of some ligands and some receptors. So these are basically all additional filtering steps that you can do. And many algorithms do this in many different ways and therefore also give a lot of different results, right? So if you run two cell-cell communication algorithms on the same data set, you actually get hugely differing results. And that's because all the tools are differing in the databases they use for ligands and receptor, the kind of way the communication score is calculated, the filtering they're applying, even the statistical tests they're applying afterwards to filter out only the relevant ligand receptor interactions. But that's basically the basic ingredients for methods that look at ligand receptor interactions. Now, if you look back at kind of our original picture with the full pipeline, then basically there are a number of things you can say about this approach. So what would you think would be kind of the pros and cons of looking just at gene expression of ligands and receptors to study cell-cell communication? You don't see the downstream effects, right? So might be that there's just expression of the ligand and the receptor and nothing functionally is happening downstream, but you're ignoring that part here because you're just looking at the expression of the ligands and the receptors. Some well-curated databases of ligands and receptors. Ideally, you would even want to know which ligands and receptors are specific to which cell type, right? That would even help you more to filter out maybe ligands that can never be expressed on a certain cell type, right? Maybe something else, if you think about what this kind of interaction is really, it's a protein-protein interaction, right? We're kind of ignoring that little subtlety here because we're looking at gene expression, at transcriptomics. It's not because the transcript is there that the protein is there, and it's not even guaranteed then that it's a functional interaction between those proteins, right? So a lot of things have to come together in order to really have a functional interaction here, right? And so these are all the assumptions that you have to be aware. So basically, all the tools we have now is very, very crude ways to estimate cell-cell communication. That's basically one of the most important messages I want to give you here. All right. This is an example of one of the widely used tools, CellChat. CellChat has the advantage that it's a quite complex modeling of this communication score between ligands and receptors because it also models multi-subunit complexes here. It does that by taking the geometric means of all the subunits in these complexes and then calculates a ligand receptor interaction activity by using some interaction dynamics, which is called Hill functions here, to calculate this interaction score here. Then this interaction score here is then filtered by doing a permutation test. And that results actually in a ranking of ligand receptor interactions here. And this is one of the typical plots you get from CellChat. This is a type of plot that you see most often in papers here, is basically showing by lines here which cell types are most often communicating with each other in your sample of interest here. So there are a lot of different tools that are all using kind of different scores, but this is just one example, CellChat here, of a tool that is very often used. Another one that's very often used is CellPhoneDB. So as I said, there are a number of pros and cons of this approach. Some of the pros is that you can do this complex modeling in some of the tools. It's simple and easy to calculate. These databases from our ligands and receptors can be curated, improved to improve the models over time. And once you have the results, you can do many downstream tasks with that. We've seen that there's also some weaknesses in the assumption here. We take gene expression as a proxy for functional proteins. We have strong dependence on the databases that we use. We don't take into account downstream functional effects. And typically, these databases are very general and not cell type specific. All these things we will see in the future will be improved, right? Then we have another big category of methods that not only look at the ligand receptor expression, but also look at the downstream effects. So that's what's called intracellular cell-cell communication models. And basically, what they are doing is also looking at these downstream genes that are affected by your cell-cell communication. And you can think that this gives you additional information. You can basically see it as a kind of footprint of your cell-cell communication event. One of the tools that is most often used in this category of techniques is NicheNet, which we developed already 5 years ago. So that was even before there was spatial omics data. But as you maybe can infer from the name, it was used to already study niche environments, right? So what is NicheNet doing? NicheNet is not only looking at ligand receptor interactions, but also looking at downstream signaling cascades and taking receptor to transcription factor kind of signaling cascades into account and further transcription factor to target gene regulatory cascades into account. And so basically this covers the whole pathway from your ligand to receptor to your whole downstream target genes, which are the genes that you typically measure in a transcriptomics experiment, right, in your receiver cell. Now, the whole idea of NicheNet is that we combine a lot of databases where we have information on all these different interactions here. And we're going to combine them all together to model this whole pathway here. And then what we're going to do is basically reverse the chain, right? So these are all kind of directed edges in a graph that basically go from your ligands to your targets. The targets are what you observe in your receiver cell. And what we're going to apply to this model is what is called abductive reasoning. Abductive reasoning is trying to suggest a cause for an observed effect, right? So the observed effect is typically some changes in your target genes due to the cell-cell communication. And what we're going to suggest is a cause, a ligand, and some receptor interaction that might have caused a change in these downstream genes, right? So that's the reasoning behind NicheNet here. So NicheNet will calculate a so-called prior model. This prior model basically summarizes this whole network in a matrix of probabilities of ligands signaling all the way down to certain target genes. And so if you measure expression of certain target genes in your receiver cell, basically you can look at changes in gene expression in these target genes due to your cell-cell communication. You can combine that with a prior model, and basically you can rank your ligands. You can see which downstream target genes they might affect, and you can also pair the ligands with potential receptors. So this is basically the approach you typically do in a NicheNet analysis. What is very interesting about NicheNet is not just a method, it's basically a very flexible framework. You can add data sources to this prior model to make the model more specific for your experiment. So what we do is we have the different steps. We have ligand receptor interactions, receptor transcription factor interactions, and then transcription factor targets. And basically all of that is kind of represented by graphs, which can be equivalently stated as matrices, right? Adjacency matrices with probabilistic transitions in these matrices. So your ligand kind of signaling data sources here, we typically improve these a little bit by using a graph diffusion algorithm, which is called the personalized PageRank algorithm. And this gives you a kind of better matrix of probabilities of which ligands are potentially regulating which transcription factors. And then the second network is all the gene regulatory data sources, which are summarizing information from regulators to target genes. You multiply the ligand receptor, the ligand regulator matrix with the regulator target matrix, and you get your ligand target matrix, which tells you what is the kind of potential for every ligand to affect all the possible target genes. You can enrich this prior model by including, for example, specific proteomics data from your own experiment or spatial omics data. You can also enrich by adding more specific edges to your gene regulatory network here. For example, you can infuse predictions from gerund inference methods like CNIC+, for example. So all of that will kind of improve the specificity of your prior model. If you just use NicheNet as it is just a very general framework, but the more context specific you can make it for your experiment, the better, of course, your results will be. And all of that is possible, right? So we also have a new version of NicheNet. We added the whole omni-path databases, many more signaling data sources as well. NicheNet can also be combined with general frameworks like Liana. There's a tutorial here. It's a collaboration with Julio's lab, where we basically give a tutorial on how to combine NicheNet, which does all this intracellular signaling, with the general Liana database. Not going to go further on that. Let's just have a look at an example of how we can use these kind of tools. So one of our collaborators is the lab of Martin Gilliams, who's very famous for studying liver and macrophages in general. And one of the questions that basically spurred the development of NicheNet was basically what defines this kind of Kupffer cell niche in the liver. The Kupffer cells are one of the type of macrophages in the liver, right? And what Martin was interested in was what are all the interactions that these Kupffer cells have with the other cells in their niche in the liver. So basically what he did was he set up an experiment where all the Kupffer cells are killed in the liver, then new monocytes come in, and these new monocytes get tissue imprinted by the liver niche and become the new Kupffer cells, right? And of course, that's a very nice model to study what signals these incoming monocytes are getting to be imprinted as Kupffer cells in the liver. So that's basically the original motivational study for developing NicheNet. So basically, our cells of interest are these incoming monocytes. We want to see what kind of signals do they get from the environment here in order to become Kupffer cells, right? And the other cells are stellate cells, L6 and hepatocytes. And basically, what NicheNet allows you to do now is do this whole cascade of modeling. You have the ligands on all these different cells in the spatial niche of the Kupffer cells. And basically, NicheNet prioritizes these ligand-target interactions here. So what you see in this inner circle here are all the target genes in the monocytes that are on their way to become Kupffer cells. And you also see the receptors for each target group here. And now you can basically see how the different cell types are kind of tissue imprinting these incoming monocytes. And you can, by looking at the target genes, you can also get some more functional insights into what all these interactions are basically doing to the incoming monocyte. So that's one advantage of these downstream signaling, that you can look at these target genes and basically see if there's enrichment in certain pathways of these target genes here. For example, you see that these stellate cells, which have interactions shown mostly in green here, they do a lot of imprinting on the monocytes to become general macrophages. Then we have a combination of the stellate and the L6 here that are doing Kupffer cell identity imprinting, all these genes here. And then we have the hepatocytes who mainly induce kind of chemoattractant and adhesion signatures here. So basically, you can see which kind of interactions have which functional effect on these monocytes as they are kind of imprinted by the tissue environment to become Kupffer cells here. We also validated these predictions. We found some new interactions here, right? Also here, these type of tools have some strengths and weaknesses. We go beyond the expression of ligands and receptors and consider downstream effects here. Also there we can improve these prior models and databases. Again, a lot of potential for downstream analysis and more kind of functional insights into how these cell-cell communications are having an effect on the receiver cell. There are also some weaknesses again, right? We don't consider any multimeric subunit approaches. We have some more complex types of analysis involving now the ligands, receptors, but also these downstream target genes. It also, again, highly depends on curated databases for all the different types of interactions now, not only the ligand receptors, but also the downstream signaling and the gene regulatory interactions. And of course, these databases are often very general and not cell type specific again.
Is there a way to, because of the layer, account for the uncertainty at different layers? Or, I mean, this depends on a lot of curated things that you might.
Yes. Yes.
Sure or not sure.
Yes. Yes. So basically, what we do in NicheNet in the prior model is we assign weights to the different data sources, and these weights are basically optimized in a kind of training procedure of the model such that we basically find which are the trustworthy data sources and which ones not to improve the model. Those were the two types of main approaches. Since then, a lot of new cell-cell communication tools have been developed. There's a very nice review paper with this nice figure here, where basically you see all the different types of models that can exist. Here at the top right, basically you have what I would call the core tools like CellPhoneDB, CellChat, which are prototypes of the ligand receptors, and NicheNet, a prototype of the intracellular modeling. Liana is a kind of general framework. I will discuss that in one of the next slides. You see that there's a whole plethora of tools for spatial transcriptomics analysis. There are also some tools for metabolite-based communication. There's a lot of new tools for comparative cell-cell communication analysis. I will also say shortly something about that in the last part of the talk. Basically, there are many tools. All the tools make different assumptions about databases and aspects of the modeling process. And there's not something like the best tool, right? Many tools model different parts of the process, and actually you might get better results if you understand a little bit how the tools are working and how they can be complementary. For example, you can combine CellChat and NicheNet. CellChat is very good for modeling the ligand receptor interactions. For example, you can first do a CellChat analysis and then work further on the ranked list of ligand receptor interactions from CellChat, and then apply NicheNet to combine with the intracellular modeling part and further refine your ranked list of ligand receptor interactions. And this way, there are many possible ways that you can combine tools. So now let's see how we can get spatial information into these tools here, right? So there are a lot of, let's say, types of spatial analysis you can do in the context of cell-cell communication modeling. So as you've seen yesterday, there are many tools that are trying to model cell neighborhoods or niches or domains, whatever you call it, right? If you have such a, let's say, niche finding tool, basically you can very generally assume that cells within that niche are interacting. That's, of course, very general, right? You just assume that the cells in a niche are interacting, but just from knowing that they're in the same niche, you don't know how they exactly interact. You just assume they're interacting. But in some cases, that can already be a very crude way of modeling cell interactions. You can have ligand receptor pairs that are ranked by, for example, any of the methods we've seen. And you can now combine that with spatial information to look at which ligand receptor interactions are spatially enriched. For example, you can do all the statistics that Mark described yesterday on ligand receptor interactions and see which ones actually colocalize, for example, okay? You can also do spatially aware ligand receptor inference analysis. Basically, that is you take the output of ranked ligand receptor interactions of any cell-cell communication tools, and then you filter out the ones that are not spatially colocalized. Very easy to do. Makes any tool a spatial cell-cell communication tool. You can do more complex things like looking at intercellular programs and intracellular signaling and incorporating, for example, the gene regulatory networks that are maybe in the sender cell or driving the communication, and in the receiver cell are putting the communication into a difference in the cell state of the receiver cell. So this is more complex types of tools. You can also look at more mechanistic models of communication dynamics. These are typically tools that are not very mainstream, but that are more based on differential equation modeling. And you can do comparative analysis of all the above tools, right? You can take a normal tissue and a tissue of a disease, and see how all these things are differing between your conditions of interest. All right. So as an example here of the fact that we have to be careful of what we call a niche, and basically a niche that's just my own definition. Here is a collection of cells that are spatially constrained to jointly implement a biological function. And that can actually happen at different levels in your tissue. For example, we study a lot the liver, which you can say is a niche, right? An organ is a niche. It fulfills a specific function. But if you look at the liver, you can see that it's a repetition of a lot of these hexagonal structures here, where you have blood coming in from the portal vein being filtered and out goes through the central veins here. And you see that even in such a basic component of the liver, there's again symmetry, right? You can take one of these portal to central triads here, and that's again a niche, right? So from the portal vein to the central vein here, you have basically a filter of different layers of hepatocytes. There's a gradient here. There's a, for example, Wnt signaling gradient that affects the gene expression of all the cells here. And for example, if you look at the Kupffer cells, the Kupffer cells are also spaced along this gradient here. And every Kupffer cell, again, has its smaller localized niche here, which was the niche that I was describing you before when describing this motivational case study for NicheNet. So if we do classical confocal microscopy, where you have the Kupffer cells in red, the stellate cells in green, the endothelial cells in blue here, basically you can see very nicely that every Kupffer cell is always paired to one stellate cell, and they're kind of spaced evenly around in this niche, and then you have the endothelial cells here. If you do 3D electron microscopy, you can even see that the Kupffer cell in red is basically hugging with the stellate cell in green, and it is having the arms kind of protruding into the veins here, because one of the functions of Kupffer cells is it's a macrophage, it's an immune cell, is to probe for things and react to things, right? But it also tells you that this is going to make it very difficult for spatial transcriptomics, right? This kind of wrapping around of cells, if you do a 2D slice, huge segmentation issues for these type of cells, right? So if we do spatial transcriptomics on this kind of niche here, so you can see basically very nicely the spatial gradient here. So you have the orange cells going from light orange to darker orange. That's basically the hepatocytes going from the portal vein, this one here, to central vein, which are smaller holes you can see here. You can see the same thing with the L-sex, right? You have portal and central L-sex, and you even see the spatial gradient on the UMAP. So basically that tells you that in some cases you don't even need spatial transcriptomics data. The spatial signature is even so clear in the normal single cell transcriptomics data, right? But actually you can study it much better using the spatial transcriptomics data. These are these kind of spatial gradients from the portal to the central vein here. This is now a spatial proteomics data set. We can do cell segmentation again on these Kupffer cells here, and we can automatically find all the spatial gradients, right? And map all the Kupffer cells onto a spatial gradient, average overall spatial gradients, and then we get a nice spatial distribution of how the Kupffer cells are arranged. And you can see there's kind of an asymmetry into the distribution of these Kupffer cells here. We can do all kind of spatial statistics on that again. So if we want to combine these cell-cell communication algorithms with all these spatial concepts, basically you can take any niche construction algorithm and then look at the cells in that niche and then apply any cell-cell communication tool onto that. What is the advantage? is basically very simple. You can control yourself the whole process. You have much more fine-grained control on what you exactly put as input to the cell-cell communication tool. There exists, of course, a lot of spatial cell-cell communication tools. Some of them are very complex, using optimal transport and whatever you want. But it's very difficult to then exactly see how the model is coming to its conclusions. Some of the simpler cell-cell communication models can actually be very nicely combined with niche finding algorithms to study your cells of interest. And so basically, there are two very general ways you can convert any cell-cell communication tool into a spatial cell-cell communication tool. The first way is what I call pre-filtering. You take any niche finding or niche construction algorithm, and then you select the cells in that niche, and then you apply any cell-cell communication tool of interest only to those cells that you know localize. The second approach is that you do a post-filtering. You use the spatial information a posteriori to adapt your ranking. So you first apply your cell-cell communication tool of interest. You get a ranking of all your ligand-receptor interactions, and then you're going to filter in that ranking for only keeping the ligand-receptor interactions that are co-localized or are part of the same niche, right? So these are very general ways of making a spatial cell-cell communication algorithm. It's often simpler than using some of these complex spatial cell-cell communication tools. It offers you a lot more control about what is happening in the process, and it's often also easier to interpret. There are also larger frameworks like Liana Plus, which do or offer you a lot of functionality of different types of analysis. For example, Liana Plus combines different things. The first thing it combines is that it's a wrapper around a lot of the ligand-receptor interaction modeling tools. The second thing is that it allows you to, for example, calculate all these local or global spatial statistics. It also is based on a previous tool that the lab of Julio developed called MISTY, and MISTY was actually a tool for multi-view analysis of your data. And basically, Liana Plus is building on top of a lot of existing tools and combines all that functionality into one big framework. For example, what you can also do is compare multiple conditions using this PyD-seq version. You can do factor analysis based on these multiple types of data. If you have multiple types of data or multiple views of your data, they typically use non-negative matrix factorization based approaches to kind of decompose your multimodal data into different factors, right? And a factor is then basically a signature that you can plot, for example, on your spatial data. And you can then have a look which ligand-receptor interactions are driving that factor, right? And that gives you kind of spatial view on your cell-cell communication events. The nice thing is you can combine different modalities. For example, this is an example from their paper where they combine spatial metabolomics with spatial transcriptomics data. And they have a brain tissue where one part of the tissue is intact and the other one there's a lesion. They apply spatial metabolomics and spatial transcriptomics data. This is combined in the Liana framework. And basically, you can see that in the lesion part here, you only have the transcriptomics part, and you basically lose this kind of dopamine effect here, which is the metabolomics part here. All right. Some of you rightfully asked the question, what about the results, right, that come out of all these tools? I think there is still a lot of work for us as a community to do. There are several reasons for that. Basically, this nice overview paper of all the cell-cell communication tools shows you that there are a lot of possible tools. And basically, anyone can make a cell-cell communication tool and publish it these days. The only thing you have to do is maybe do some variations of a combination of existing techniques. You cherry pick some data sets on which your method gives good results, and you publish that, right? But nobody really validates the performance of the tool. And one reason for that is that we have very few data sets where we have ground truth to objectively and quantitatively evaluate the performance of cell-cell communication models. This was a situation that 10 years ago we had in a very similar problem. Maybe you know pseudo-time inference or trajectory inference models. So 10 years ago, we were in that same situation there. We had a booming of a lot of tools, and nobody really quantitatively performed the benchmarking of those tools. We did a huge effort at that time on doing a benchmarking, generating real and synthetic benchmarking data sets, defining quality measures, and basically that was a huge contribution to the whole community. And that benchmarking paper is still one of the most cited papers in the field. But in the case of cell-cell communication, we don't have sufficient ground truth data yet to do a really comprehensive benchmarking. Nevertheless, you can try to compare the output of different tools. This is the Liana paper, where they did just a comparison of the output of the different tools, right? So either you take the same resource, so the same database, for example, for the ligands and receptors, you run all the methods with that same database, and you check the concordance of the results. That is surprisingly low, right? Even if you use the same database. You can also use the same method with different databases and then see how does that compare, and that is also surprisingly low, right? So this is the conclusion from the paper. So very, very low pairwise Jacquard indices, which is the overlap between the top ranked predictions of all these tools here. So it's something to cry about, maybe, but it in any case indicates that there's a lot of potential for improvement to advance the field further, right? Some of these low performances also have to do with choices the models are making, not always the appropriate types of data that we actually need. This is another comparison. In this Liana Plus paper, they also did a comparison of the output of different cell-cell communication tools, and now they validated them using co-localization, right? They say if a ligand and receptor is co-localized, then actually we assume it's a proxy for interaction. And also you see that the performances are still low, right? An AUC of 50 is random guessing, right? So you see that many of the tools some are even doing worse than random guessing, right? A lot are doing slightly better than random guessing. In the NicheNet paper, we did a slightly different evaluation of our framework. We collected 111 transcriptome data sets where a particular ligand was added as a treatment. And what you can then do is compare the untreated with the treated sample, see what is the difference in gene expression, and see if NicheNet can correctly predict which ligand was added based on the transcriptomics data, right? You can also do the other way around. You can start from the ligand that was added as prior knowledge and see if NicheNet can predict the affected target genes. So those are the two types of validation you can see here. The first one here, the first graph, is how well you predict the affected target genes when you know that a certain ligand is added. Gray one is kind of a model where you just have randomized edges in the model. The different blue ones are the different versions of NicheNet with either an unoptimized or this weight optimized network. The weights, as I said in one of the previous questions, are basically to determine the confidence of the different databases that we use. You see that the optimized weight model is basically performing a lot better. And then the second graph what you see here is how well NicheNet is able to correctly predict which ligand was added, only based on the transcriptomics data. And you see that that's much higher, right? We have almost something like 90% accuracy in predicting that ligand in the top part of the ranking. So that's quite good news, right? The good news is that more and more assays are appearing that are able to generate ground truth interaction data sets. This is a very nice paper, the immune dictionary paper. This is an assay universal lipstick that basically allows you to measure in vivo cell-cell interactions. We can imagine that those types of assays will kind of emerge more and more and give you some way of either validating the model or basically just avoiding to have to resort to predictions. This is just another way of validating, but then more in the dynamic setting here. So this is a 2-photon imaging data set where we have these Kupffer cells and these monocytes. I don't have the time to show you the whole movie, but basically the Kupffer cells in red are dying and the monocytes are kind of taking their place as the new niche cells here. So I'm basically going to conclude the lecture by showing you what's the next kind of things in the field. And one of the most interesting directions is comparative analysis. So for example, you have a cohort of patients, maybe some of them are responding well to a therapy, some of them are not responding well to a therapy. Can we basically see what is different in terms of cell-cell communication between the responders and the non-responders? For that, we basically developed multi-NicheNet. Multi-NicheNet is a multi-sample, multi-condition version of NicheNet that allows you to really model complex designs and correct for batch effects and other covariates, offers a lot more flexibility in the prioritization scheme. Basically, in this new prioritization scheme, you can, in a weighted fashion, combine different criteria. One of them could be spatial colocalization, others could be cell type specificity, and so on, right? And we also developed a lot of new visualizations. I'm not going to go too much into the details here, but basically this would allow you to compare, for example, different niche environments. You take your tissue, you have different niche environments. Maybe if you think back about this tumor example where you have different types of tumor growth, you want to see which interactions are different between that. You can do that with multi-NicheNet. You can again see which interactions are maybe specific for a certain condition. You can compare that across conditions. We have a lot of new types of visualizations for that. You can also model all the signaling cascades. In this case, we had time series data before and after treatment. So you can also have a look at the dynamics of the cell-cell communication, and you can see which interactions change over time, right? After the patients are treated. It's all in the preprint. You can have a look at that. Just going to end with other types of models that I think are very interesting. And these are dynamic models based on differential equations that really either try to model more, for example, the niches by looking at the diffusion of the ligands and how certain sender cells are producing these ligands, receiver cells are consuming these ligands, and you can actually model the whole response curves of that, or more specific models where you try to unravel, a whole pathway, right? You try to measure as much as possible, maybe using combination of transcriptomics and proteomics data over time, and you try to model all the responses of this communication process. So this is an interesting paper. They model 11 protein components using 1,000 ordinary differential equations and 70 kinetic parameters. You can imagine you can maybe also in the future have more machine learning-based models that try to model these interactions. All right. And so by that, I'm concluding and thanking all the labs and all the collaborators that collaborated with us, and especially Robin, who did all the basic NicheNet and multi-NicheNet developments. And I hope we still have a bit of time for questions.